I'm now my great pleasure to introduce the 2015 Canada Gairdner Wightman Award winner. And I'll quote from the rules of the game here. The Canada Gairdner Wightman Award is given annually to a single individual who has displayed superior leadership in a specific field of biomedical science or sustained contributions at an international level, and institutional and scientific leadership in Canada in advancement of biomedicine nationally and internationally. I think to me, and I think it's pretty obvious, that Janet Rossant is the perfect exemplar of the Wightman Award. She is internationally known for her outstanding research on developmental and stem cell biology, in particular her work on the Trophoblast, her early work on the Trophoblast, on vasculogenesis, on developing new technologies of mouse mutagenesis, and more recently her work on embryonic stem cells has been widely and deservedly recognized around the world. She's also led the Research Institute at the Hospital for Sick Children, Canada's largest research institute, with great distinction, and she stepped down from that role a few months back. Nationally and internationally, Janet has served on many national and international advisory boards and committees, and she's very much in demand as an advisor, I think because of her clear thinking, her ability to articulate a clear way through a very complex and difficult situation. She has received many honors and awards both in this country and abroad for her work and for her contributions to science. Most recently, other than this one, she was made a Companion of the Order of Canada. I first met Janet as around the first time I first met Lynn. Dennis Azar, then a postdoc in my lab, and I were trying to develop retroviruses as vectors to introduce genes into the mouse germline. Janet was then at Brock University, south of here. And so we started looking for someone who knew about how to work with mouse embryos. There was really only one person, I think, in this country who knew how to do that, and that was Janet. And so we started a collaboration that lasted for decades. Janet was a superb and fun collaborator, and when the new Mount Sinai Hospital Research Institute, now the Lunenfeld-Tannenbaum Research Institute, started, I called her to see if she would be interested in coming to Toronto. To my great delight, she said yes. Since then, Janet and I have been close colleagues, we've been collaborators, and I'm also very proud to call her a close friend. Janet.
Thank you, Alan, for those very kind words. It is, of course, an honor to receive this award for myself, for my lab mates, and all the colleagues and collaborators I worked with over the years. And I'll mention some of them as we go through today. But what I wanted to do today was to explain to you why I like the little embryo you see on the picture here. I do wish we had better lighting. It's beautiful. This is the mouse blastocyst. And today I'm going to try and tell you a story about how working on the mouse blastocyst over many, many years has led off from my work and many people's works into other important areas, including, of course, stem cells. So what are the questions that have driven my career? I started working on the blastocyst when I was a graduate student in the UK, and I wanted to understand how, at this stage of development, where the embryo has only actually made 3 cell types, it's the size of a speck of dust. This is a mouse blastocyst, size of a speck of dust, 100 cells, it's tiny. But from the time of making the fertilized egg to the blastocyst, it has made some decisions about what it wants to do. There are clearly 3 distinct morphological cell types. We now know, with lots of pretty molecular markers, as you see here, that they are molecularly distinct cell types. But we didn't know what they produced or how they got there. So those 3 cell types were named the trophectoderm, the outer cell layer of the blastocyst, and at one end of the cavity here, we see this group of cells called the inner cell mass. Unlike Drosophila, mouse people have very boring names for everything. Inner cell mass. And inside the inner cell mass, we have this little group of cells called the epiblast, and a layer of cells on the surface called the primitive endoderm. So what I wanted to know was, well, we've made these 3 cell types. What do they give rise to? Are these distinct lineages? Are they restricted to particular cell types in later development? And if they are, when is that restriction occurring? And more importantly, how is it that they get established from a single cell, the egg? And with the limitations of knowing that this is a tiny embryo and we can't get very many of them, very early on we wanted to know if we could make stem cell lines from these 3 cell types so that we would have a large amount of cells to really move from the cellular to the molecular level. So what I want to do is to explain to you why those fundamental questions matter. First of all, I do believe that the early mouse embryo is really one of the best systems to understand fundamental cell fate specification. It's very simple. You can grow the embryo in a fully defined medium, fully defined culture medium, in a petri dish. You can put it back in a mouse and have it complete gestation. So you can manipulate that embryo in any way you want and test the effects later on. And also, it really is a slow and easy to monitor process. We can do videos of the embryo, we can study the whole process in the culture dish. So it's becoming actually more and more a popular system to really understand cell fate specification at a single cell level. So that's our fundamental question too. And if you think about it, what we want to understand is this process here, whereby we undergo what we can call a totipotent to pluripotent transition. We start with a fertilized egg that makes everything. Early cleavage stages, the cells divide, but the cells have not made any decisions. All these cells are totipotent and they can produce all the cell types of the later conceptus. But as development occurs and cells start to generate an outer layer of cells and an enclosed group of cells, so that by the early blastocyst stage, we have this outer layer of trophectoderm and an enclosed group of inner cell mass. And what we know from a lot of work that I'm not going to tell you about is that it's actually these epiblast cells that are the pluripotent cells of the blastocyst and produce the entire fetus. So we go from totipotency to pluripotency. And the question is, how? First of all, we do know from lineage studies from my lab and many labs over many years exactly what those cell types give rise to. The outer cells, the trophectoderm, form the trophoblast layers of the placenta, incredibly important for a mouse embryo to survive in the uterus. The primitive endoderm cells on the surface of the inner cell mass are also extra embryonic. They make endoderm layers of the yolk sac. And as I just said, it's only these pretty pink cells. They're pink because they're stained to an antibody to OCT4, which is a famous pluripotency factor. These cells produce the entire fetus. So we knew that from our chimera studies and lineage studies many years ago. And we also knew from manipulation that by this stage, fate was fixed. You can't change cell fate. And in fact, we're still trying to explore exactly the timing of when cell fate is restricted leading up to this blastocyst stage. So we knew, we've answered question number 1, we know what they give rise to. We know pretty well when fate is restricted, though again, as we get in more detail, we're still pursuing that. Now we want to know how that happened. How did we go from cells that all were totipotent to these 3 cell types? And this is where it's taken us a long time, but just in the last few years, we're beginning to really move from understanding this at a morphological level to a cell signaling level. So I'm now going to jump from all those years ago when we defined cell fate to today, when we now, and give you a little update on what we know about how those cell types are actually established in the mouse blastocyst. And it's summarized in this diagram here. Here's our cleavage stages again. We're going through from these totipotent cells to the blastocyst stage with the 3 cell types. And what we know is that between the 8 cell and the 16 cell stage, the outer cells of the embryo become polarized, and enclosed cells begin to look different from the outside cells. These polarized cells are polarized by all your favorite PAR proteins and everything else that's involved in making a polarized epithelium. The enclosed cells, however, show a different phenotype. And what we now know is that these inside cells are in the inside cells, the Hippo signaling pathway is activated. And activating Hippo in the inside prevents the turning on of the genes that make the trophectoderm on the outside. And I'll show you a bit of data, more recent data, to support that in a minute. So we have a positional event with a difference in signaling between the inside and outside cells that sets up the inner cell mass in the trophectoderm. Then how do we make these inside cells turn into the two-cell layers of the blastocyst? That turns out to be a different process, not a positional dependent one, but one in which stochastic activation of FGF-ERK signaling in individual cells in the inner cell mass is translated into cell fate and cell surface differences between the cells that cause the primitive endoderm and epiblast to segregate out into the layers of the blastocyst. So it's a connection then between morphogenesis, which we've known about for many years, and signaling pathways that leads to linear specification. So let's just look at a little bit about what we know, some of the more details of that. So what is this Hippo signaling? So how come we knew about the inside-outside differences in the mouse blastocyst, actually first proposed in 1967 by Tarkovsky and Wrobleska, and it's taken us this long to work out what signals are actually different between the inside and outside? Well, it took that long perhaps because it turned out to be a signaling pathway which we haven't known about for that long. Not one of the classic ones, the FGFs, the WNTs, and everybody else, but the Hippo signaling, first identified in Drosophila as a growth control pathway, rapidly shown to be conserved in mammalian cells, and to be very much involved in growth control, such that components of this are often involved in tumorigenesis. And in collaboration with Hiro Suzuki's lab in 2009, we showed that, in fact, components of this pathway are playing a different role here and are specifying cell fate. And in particular, this serine-threonine kinase, LATS kinase, which is the key kinase in the Hippo signaling pathway, what it does is it phosphorylates a co-activator called YAP. And when YAP is phosphorylated, it stays in the cytoplasm, cannot enter the nucleus, and bind to its transcription factor partner, in this case TEAD4. And YAP and TEAD4 are required to turn on CDX2, which is a trophectoderm specification gene. So in the inside cells, Hippo signaling is activated, and we see that YAP is excluded and no CDX2 expression occurs. In the outside cells, we see that there is no Hippo signaling active, YAP enters the nucleus, CDX2 turns on, and we continue down the pathway. So this opened up then the possibility of explaining a bit more about how this occurs, but of course it leaves lots of questions like, well, why is Hippo signaling activated in the inside and not the outside cells? We know it's activated by cell contact mediated events, but all the cells are in cell contact. These cells are polarized, and in cell cultures that's often associated with active Hippo signaling, and yet not here. So what's going on? Well, my lab and Hiro Suzuki's lab and a number of other labs now have started to look at other components of this pathway and try to ask exactly what is upstream of activating that signaling in the inside cells. And many groups have shown that the serine-threonine kinase is involved in a complex that involves many other molecules that are required to activate LATS activity and phosphorylate YAP. And in particular, Merlin in Drosophila, or NF2 in mammalian cells, and angiomotin are shown in mammalian cells, particularly by DJ Pan's lab and Hiro Suzuki's lab, to be absolutely necessary for this LATS activity to be associated with the cell membrane and to be activated by upstream cell contact mediated signals. So what we wanted to know is whether the same things was happening in the embryo. Hiro Suzuki's lab showed that angiomotin was involved, and my lab looked at Merlin or NF2. And this was work by a graduate student, Katie Coburn, when she was in the lab. And she showed that NF2 is in the early embryo and is actually maternally inherited. The protein is laid down during oogenesis, and you see large amounts of NF2 ready to go to help Hippo signaling in the embryo. So in order to find out whether it's required for this process, what we had to do was actually to knock out NF2 both maternally and zygotically, which you can do with CREAD drivers that are expressed in the oocyte. And what you see when you do that is these are wild-type embryos on the left, showing that in the inner cell mass, YAP is excluded from the nuclei and is present in the nuclei in the outside cells. So this is where Hippo is active. If you remove NF2, then all the inside cells show nuclear YAP, just like the outside cells. And indeed, all the inside cells then turn on the downstream gene, CDX2. Haha, there's always something when you go from a Mac to a PC. I missed that. That's YAP. This is CDX2. So what you see then is that essentially you've still got a blastocyst on the face of it, but now you've changed cell fate, and all the inner cell mass cells have turned into trophectoderm cells. So this has really then led us to suggest that many of the components of the Hippo signaling are indeed required and active in the inside cells. So the current model for what's going on here is that in the normal embryo, NF2, angiomotin, all around and associated with LATs at the membrane in the inside cells, causing phosphorylation of YAP. But in the outside cells, NF2 is still there, but angiomotin, and this was work from Sazaki's lab, is not able to associate with NF2 and activate this pathway. So we get YAP and we get trophectoderm formation. If you knock out NF2, then essentially you don't get this complex in the inside cells either, and all the cells produce CDX2 and produce trophectoderm. Polarity is actually important to segregate out NF2 and angiomotin, because if you interfere with polarity, then what happens is you actually get this complex formed in both inside and outside cells, and now all the cells think they're inner cell mass. So we're beginning to get there. There's lots more details to work out, but it does suggest then that really Hippo signaling is what's driving inner cell mass trophectoderm segregation in a very interesting way. So that's number one. We've got this combination of cell contact, cell polarity, and Hippo signaling driving that first cell lineage event and driving then this first movement away from totipotency to pluripotency. But what about the next phase? If we really want to get to the pluripotent epiblast, we have to stop cells making primitive endoderm as well. So how do we distinguish primitive endoderm and epiblast? How does that occur? Well, that became a little clearer, again, more recently. We had originally proposed that the primitive endoderm probably also formed by some kind of position-dependent event. After all, it's sitting on the surface of the inner cell mass. But as we got better and better markers for these lineages and understood more that this event was a progressive event, what we showed, and this was work by Claire Chauzeau when she was a postdoc in the lab, was that if you look in an earlier inner cell mass, you don't see this spatial distinction between the layers. And if you look at GATA6, which is a primitive endoderm marker, or NANOG, which is an epiblast marker, they're expressed in a mosaic within the inner cell mass. And it's not then until you get to this stage that they appear to segregate out. So what happens to cause this mosaic to begin, and how do they segregate out? Well, various numbers of experiments, starting actually with that work that Claire did in collaboration with Tony Pawson's lab, led us to suggest that what was going on here was the importance of the FGF signaling pathway, and in particular the downstream activation of the MAP kinase pathway or the ERK pathway. And cutting a long story short, what we think is happening now is that within the inner cell mass, individual cells are responding to the local level of FGF-ERK signaling. And if they have high ERK signaling, they become primitive endoderm. And if they have low ERK signaling, they become epiblast. And so if you manipulate this by, for example, blocking with a small molecule, blocking FGF-ERK signaling, then all the inner cell mass cells become epiblast. You don't make primitive endoderm at all, still the normal number of cells, but you've changed cell fate. This is not just permissive for making primitive endoderm, because you can send all the cells the other way. If you stimulate FGF-ERK signaling, the same thing happens, only this time all the inner cell mass cells become primitive endoderm. And you can obviously play around in between and regenerate the kind of mosaic picture I showed you before. So really then what we think is happening is that this is a kind of stochastic event. Individual cells begin to upregulate FGF4, and they'll talk to their neighbors who express the receptors, and eventually you'll start to get a sort of amplification event that the cells that respond turn on the downstream genes that specify primitive endoderm, and the cells that express FGF4 don't turn on the downstream genes and stay as epiblast. Downstream of these transcription factors, there are cell surface molecules that differ, and then eventually the cells segregate out and no longer talk to each other, and we have restricted lineages. So, by studying the fundamental pathways of making the blastocyst, we've gone from cell lineage to cell signaling to understanding where pluripotent cells come from. And also, much more importantly originally for me, was understanding where the placenta comes from. So we've been very interested for a long time in the trophoblast cells because they are absolutely mammalian specific and required for a mouse embryo or a human embryo to survive. And I always say that the most important structure is the placenta. None of us would be here today without the placenta. So, with that in mind, we had a long interest in being able to not just understand how these cells form, but understand how they're maintained and be able to generate cell lines that would enable us to study in more detail, particularly the trophectoderm and the trophoblast. So we wanted to know whether we could derive lineage-specific stem cell lines. And starting with the trophoblast, we indeed were able to show that we could derive trophoblast stem cells, which we and others have used a lot to study trophoblast development and pregnancy disorders. So how did we get to trophoblast stem cells? Goes back to FGF again. The same signal that's produced by the epiblast cells that leads to this segregation of epiblast and primitive endoderm, when they're not talking to each other, the signal from the epiblast hits the overlying trophectoderm and causes these cells to proliferate, activate ERK signaling, turn on downstream transcription factors, and become trophoblast stem cells, which continue to proliferate as the embryo implants in the uterus and form this region here called the extraembryonic ectoderm. With that information in mind, Satoshi Tanaka, when he was a postdoc in the lab, was able to translate that discovery into forming permanent mouse trophoblast stem cell lines in culture by taking blastocysts or early post-implantation trophoblasts and growing cells in the presence of FGF4 and conditioned medium from fibroblasts. The pathway there is we now think is nodal signaling. And then these cells retain the capacity to proliferate indefinitely in culture, self-renew, and when you take away the signals, they differentiate into all the specialized cells of the placenta. Importantly, they really are stem cells. They really do remember where they came from. They recapitulate the trophectoderm lineage in chimeras. So if you take those trophoblast stem cells and put them back in a blastocyst, they don't contribute to the fetus, because that's not what they were committed to do, but they contribute to all cell types of the placenta, as shown here with GFP-expressing cells. So they have continued to be something we've worked on and now are trying to replicate in the human system. But actually, although I love trophoblast stem cells, they're not the most famous stem cells from the mouse blastocyst. Can we get other stem cells from the mouse blastocyst? Yes, we can. So we made trophoblast stem cells. We've also made what we call Zen cells, which are cells from the primitive endoderm. But the most famous stem cells from the mouse blastocyst, of course, are embryonic stem cells, which arise from the epiblast progenitors. And because the epiblast we had shown is capable of making a whole mouse, the capacity to be able to replicate and maintain those cells in culture has, of course, been extraordinarily revolutionary. So here are embryonic stem cells derived from the embryo, expressing specific lineage transcription factors like OT4 and NANOG. And importantly, when you put those back in the blastocyst, they contribute, as shown here, with Z-expressing cells, to all cells of the fetus, including the germline, but they don't make the placenta, and they don't make the primitive endoderm. So they are pluripotent and not totipotent, whereas Zen cells make the yolk sac, trophoblast stem cells make the placenta. So there really is a lineage distinction. So embryonic stem cells were first derived by Martin Evans and Gail Martin independently. Martin Evans shared the Nobel Prize in 2007 with Smithies and Kopecky because mouse embryonic stem cells really allowed a revolution in mouse genetics. And the revolution in mouse genetics came about because Kopecky and Smithies showed that you could target mutations to any gene in the genome in an embryonic stem cell using homologous recombination. And because these cells can recapitulate development, you could get a change in a gene from the culture dish through a chimera and into the germline. That really then opened up the ability to manipulate the mouse genome, which you've already heard about in many of the talks today. We all talk about knockout mice as though it was just something we all do and it just happens, but it really began because of embryonic stem cells. So we were actually early players in this game too. Because we could make chimeras, because we were interested in development, because we had embryonic stem cells in the lab, we had a really amazing time at the Lunenfeld Research Institute, particularly when I was collaborating with my colleague Alex Joyner, who was there at the time and now is in New York, her graduate student Bill Scarnes, my postdoc Achim Goslar. And at that time, 1989, we published 2 papers. First, one of the first mutations in embryonic stem cells in a developmental gene in which you could target a gene that's not selectable in cells in culture. And then secondly, and this was really the invention of Bill Scarnes, developing ways not just to target genes in embryonic stem cells, but to develop what we call gene trap vectors, in which we could mutate and identify novel developmental regulated genes. So this was the beginning of a very exciting time. So we continued, as many people have, to use embryonic stem cells for mouse genetics. The embryonic stem cells have really revolutionized mouse genetics, and it's only now that we're seeing perhaps a switch from embryonic stem cell-derived mouse genetics to CRISPR-Cas, which is going to be the next revolution in genome editing, which you're going to hear about tomorrow. So we knew that we could use ES cells for mouse genetics and making models of human disease. But I was still intrigued by this concept that in the mouse, when we take embryonic epiblast cells, we can actually show they make a whole mouse. So we wanted to know, were ES cells really like epiblast cells? Are they truly pluripotent? Can they make a mouse? And so this was a collaboration with my, again, long-term colleague and friend Andras Naj, and here we are up the top of a very high building in Hong Kong a couple of years ago. And what Andras realized was that we could play a trick. Knowing what we know about the embryo and about stem cells, we knew that embryonic stem cells can contribute to the entire fetus, but they don't like making the placenta and extra embryonic tissues. And we knew from the literature that if you made an embryo tetraploid, just double the amount of DNA, you could make an embryo that made trophoblast and endoderm, but it wasn't very good at making epiblast. So we figured this would be a complementation system, such that the only way anything could survive is with the ES cells taking over the fetal compartment. And that's what happens. Here you see a blue fetus derived from these blue embryonic stem cells, supported by the tetraploid placenta. So we showed we can make fetuses. Can you make mice? And as we got better and better embryonic stem cells, the answer is absolutely yes. You can take cells that have been many generations in a petri dish, and using this trick, you can generate whole litters of GFP expressing mice, and these are perfectly normal. They'll grow up, they'll breed, and they are absolutely normal mice. That is fundamentally important when you think about translating stem cells from the mouse to the human. Because if we are going to take this power of pluripotency and generate human cells that we might want to put back in humans to repair disease, we better know that those cells are truly pluripotent and that it's possible to keep them in a normal developmental state over generations in the petri dish, because that's what you're going to have to do. So that background then, that idea that ES cells make a mouse, really was very important as people then moved on to generate human pluripotent cells. First, human embryonic stem cells derived by Jamie Thompson, and then induced pluripotent stem cells derived by Shinya Yamanaka, in which he was able to actually replicate the process of taking adult cells and turning them back into cells that behave like the early embryo by knowing the genes that specify epiblast cells in the embryo itself. Now, these patient-specific pluripotent cells that Yamanaka developed have really changed the whole scene, because now you can make stem cells from everyone. You can model human disease in the petri dish. You can study disease. You can do drug testing. You can do toxicology. And as we move into regenerative medicine, both embryonic stem cells and iPS cells become incredibly important sources of cells to treat degenerative diseases. So here is Yamanaka, who of course received the Nobel Prize with John Gurdon for this work, but here he is in Toronto, that's me standing next to him, receiving the Gairdner Award here in Toronto. So Gairdner again recognized Yamanaka before the Nobel Prize. And so, a revolution in mouse genetics, but embryonic stem cells don't just make a mouse. Human embryonic stem cells then are also pluripotent and open up new frontiers in regenerative medicine and modeling human disease. So where do we fit into this part? Well, when I moved to sick kids, we knew that we could begin to make embryonic stem cells. Soon after that, pluripotent iPS cells came on the scene, and very early on, we started to generate iPS cells in collaboration with a number of groups at Sick Kids and elsewhere from kids with disease. And so what we now are interested in is asking whether we can apply our knowledge of stem cell and developmental biology to understand and treat childhood disease. And in particular, we've focused on cystic fibrosis. Cystic fibrosis, as you know, is a rare disease. It's caused by mutations. It's recessive. It's caused by mutations in the CFTR gene, famously cloned at Sick Kids in 1989. And it's been, over the years since 1989, the life expectancy for CF has improved dramatically. However, all those improvements have come about by treating the treatments and not the underlying cause of the disease. In the last few years only, we've started to see new drugs coming through to the clinic that really are aimed at fixing what's wrong with the protein, the channel protein that goes wrong in CF. And they're starting to come to the clinic. However, those treatments are very expensive. They don't work on all patients. There are many different mutations that underlie CF, but even if you have the same mutations, that different patients will respond differently to the drugs. So what we would like to be able to do is to really develop some means of rapidly assessing which patients are going to respond to which drug, and also develop assays in which we could develop new drugs to treat some of the mutations for which there are currently no molecular targets. So what did we do? So what we decided to do then was to make iPS cells from patients with cystic fibrosis and see if we could direct them towards the different cell types that are damaged in CF disease. And my lab has worked on trying to generate lung epithelium from iPS cells. And this was work largely by Amy Wong, a research associate in the lab, collaboration with James Ellis, Christine Baer, and Phyllis Ratjen in the CF group at Sick Kids. And again, without going into any of the details and cutting a very long story short, what Amy has been able to do is replicate the developmental pathways that occur in the embryo that take pluripotent cells first through to endoderm cells, and then through a series of steps of growth factor pathways, and eventually putting these cells onto an air-liquid interface that replicates how lung epithelial cells grow, transforming iPS cells into ciliated lung epithelial sheets that we can use to study the disease itself and also begin to test drugs on these patients. So here's just a proof of principle that we can use this iPS cells to detect the extent of response of different drugs. So here we are again. Here are the lung epithelial cells expressing CFTR. These are normal embryonic stem cells differentiated to lung epithelium. And you see CFTR is membrane associated. The most common mutation in cystic fibrosis is delta F508, which is an in-frame deletion, which causes the protein to fail to be processed through the ER. It also, when it does get to the surface, it's still not functional either. That's a different story. But you see here in these CF-derived patient iPS cells, they make lung endoderm, but there's no CFTR on the surface, and it's in these blobs inside the cell. When we treat it with one of these putative drugs for treating this mutation, then we see that we can show CFTR moving again to the cell surface. So the cells are responding to the drug, and we can measure this response not just by immunofluorescence, but in Christine Baer's lab now in a quantitative fluorescence assay. So we're moving this from a large scale to a multi-well format where we hope to be able to screen large numbers of patients and screen large numbers of drugs and begin to define exactly the right patient for the right drug at the right time. Not just with lung cells, but across Toronto we have people also exploring the use of other cells you can derive from iPS cells, like cholangiocytes. We use nasal epithelial cells, pancreatic duct cells, all of which give us a new way of measuring and understanding CF and how to treat it. So what we want to do then is to take patient skin cells, reprogram them to iPS cells, and differentiate them into lung epithelium. We're doing that on a small scale. We're going to scale it up. We're also interested and trying, with some success, to cut this step out. It's nice to go back to the beginning of development, but if you have to take them all the way through differentiation to get to lung, isn't there a quicker way to go from skin to lung? And we're looking at that as well. So And what we have now is a large consortium. Just recently, last week, there was an announcement that between CF Canada and the Sick Kids Foundation, there's now a fundraising drive to establish the program in individualized CF therapy, where we will use these different techniques to enable precision medicine for CF patients across Canada and around the world. So, there you are. Why does the blastocyst matter? Fundamental questions right through to using iPS cells to study and perhaps treat disease in new ways. But it's circular. We've got to go back. We continue to go back. We're always going round this circle to develop, understand better how this occurs, how to get better pluripotent stem cells, how to understand pluripotency, how to understand development, and how to use it to treat disease. So this is all about teamwork. Over the years, this is about teamwork. My lab, my many lab members, this is a recent lab reunion. Amazing colleagues, particularly colleagues at Samuel Lunenfeld Research Institute, and all my colleagues, all the faculty, trainees, and staff at Sick Kids, where I've had the privilege to lead the institute over the last 10 years. And it all begins and ends with the blastocyst. Thank you.